Okay, so what was the hypothesis for that little video? What? Can or ants do count. Ants count. Okay. And the predictions were that if you manipulated the ants by changing their leg size, they should have trouble initially finding their way home because they can count, but they're counting longer, shorter steps. So you're actually testing those predictions. Now, okay. There's 3 ants, one of each kind. Is this a good experimental design? What do you think? Just testing one subject. Let's say you want to find out about vitamin C in colds. You give one subject vitamin C, you give the other one not vitamin C, and you infect them with colds. Will that tell you anything? Why?
I was going to say for different reasons, I think kind of prompted is because they only tested it once from a child. They didn't see the variations.
Exactly. Not all ants are created equal, nor are humans created equal in terms of their immune systems. You tested one person's immune system, and they're immunodeficient, let's say they've got AIDS, and you gave them vitamin C doesn't work, but let's say it did. They would probably still get a cold because they're immunocompromised. So, there's variation. Variation in subjects, in this case, ants. So, to fix that, you need what's called a large sample size. So, you have a group that don't have their legs cut off, but they're or lengthened, but they're still handled by the experimenters. Then there's a group who lose their legs and the other ones who gain longer legs. So there's a control group in that, and that's the one who's had their legs just, and nothing has been done to them. Now, what you want is you want what's called a representative sample of the population. If you only use, like we said, one ant or one person in an experiment, you wouldn't capture that variation, and you might get an individual who's not representative of the whole population. So, in fact, the bigger sample size is better. Plus, we use statistics, which we'll get to, but we use that to detect if there's a difference between the 2 groups or 3 groups. So in the ants, there were 3 groups. A placebo or a control group, and then 2 treatments. Vitamin C, 2 groups, control, and then a person who gets vitamin C. And with the ants, they know who they are, what treatment they got. But with the vitamin C, we don't tell the individuals whether they actually got vitamin C or they got a placebo pill that looks just like vitamin C. and they're randomly assigned to each group. In other words, they're randomly assigned to either getting a pill of vitamin C or not getting a pill of vitamin C. And then if there is an effect, in this case with the ants there was certainly an effect, if you have a big enough sample, you're more likely to detect the difference. Okay. Now, one thing first. I said representative sample, and this is very important. Starting about 30 years ago, women who entered menopause, if you don't know this, women who enter menopause have a bunch of side effects. They get hot flashes. Looks like they get some bone loss. It seems like they get maybe more cancer. And so what they did was, over time they thought, well, if we replace estrogen, we give women estrogen, maybe the hot flashes will go away, yeah, and they'll be protected, let's say, against cancer or things like that, or bone loss. And so they did. And then over time, they did surveys of people, of women who in fact got estrogen and those who didn't. They didn't assign getting estrogen or not getting estrogen randomly. They just did a survey. And they looked at the number of people who, let's say, had heart attacks, women who had heart attacks, so the number of people who suffered significant bone loss, whatever that is. And, in fact, they found a difference. So it looked like estrogen actually helped women. No one actually ever did a real experiment like we just looked with the ants. In other words, you take a group of women and then you randomly assign one woman to get either a shot of estrogen or a shot of saline solution. And the women don't know which one they got. And over time they found out, in fact, getting estrogen was actually worse. Higher incidence of heart attacks, higher incidence of estrogen-sensitive tumors, help with bone loss. And they're going, what happened? So the original survey, which group of women do you think had access to medical care more often? The group that said they got estrogen or the group that said they didn't? Anybody? What do you think? Well, just think about it. We have a survey. People said they got estrogen, and then another group of people that they sampled said they didn't. Which group do you think was more likely to have seen a doctor over that period of time?
Once it did.
Yes, exactly. Well, it turns out that those women and that population, that subpopulation, actually they were wealthier, they had access to good health care, and they were generally overall more healthy than the other group. They didn't have access to health care and didn't get estrogen replacement therapy. So what the original survey showed was misleading because it was not testing estrogen, it was actually testing socioeconomic status and health care. Now when they actually did the experiment, like I said, they randomly assigned women to either one group or another, either getting estrogen or being the control, getting a placebo, like saline solution. And they were actually able to see that there was actually a negative effect in terms of estrogen. Now, science, like I said, scientists are the same as everybody else. They do all kinds of foolish things like confirmation bias. But eventually it came out, right? So the ant thing was an example of what we call a good experiment. And we're going to go through why. Oh, secondly, and German. What was the problem with that lab? Anybody? What was it? Tell me what gender the scientists were. Men. Why is that? Is it because women can't do science? Or is it just because men are stupid? What do you think? Men are stupid. Okay. So, luckily now, there are more and more scientists, women scientists out there. Or, in the health profession, more and more women doctors do everything from general practice to surgery, all that kind of stuff. And there are women who get a PhD in something, and they go on and get their own labs and they make discoveries. Some areas of science are more likely to have more women than others. Biology, there are certainly more women in biology than there are in physics, or chemistry, or geology. But it's slowly changing. It used to be thought that, oh, brilliant mathematicians are men. For some reason they do it better. And not so much. Now, I got through calculus, but I have a limited ability in terms of mathematics. And I frankly know lots of women that are much better mathematically inclined than I am. So, things are changing, but I just want you to understand this is still a leftover holdover from previous years in gender discrimination and all that. Whether people are aware of it or not, in terms of like accepting grad students. Okay, now we talked about trying to find out what's actually true. And one of the parts of testing a hypothesis, what's the first thing? What do you think?
You have to observe.
Observe it, right. And you're walking around and you look at these ants and you start thinking to yourself deep, dark thoughts about cutting off the legs of ants, right? So you have an observation and you want to test that. Then what's the next step? What's the next step? After you observe something and you want to find out what's going on. Not experiment yet. Not quite. What do you get the prediction from? Starts with an H. Hypothesis. Hypothesis is an explanation. That's all it is. Now you try to make good hypotheses that actually make, like you pointed out, predictions, specific predictions, where you can determine there's a difference in terms of doing an experiment and a treatment effect, like with the ants. There was a prediction about how the ants would move and whether they could get back home or not. That was a prediction from the hypothesis that ants count. Now, remember, we're not talking about ants that are sitting there consciously thinking 1, 2, 3, 4, no. But there's something in their brain that actually remembers things, like how many steps they take, and you can fool that system. Okay? And then, finally, you have these predictions, and you go and you test them. You test the predictions of the hypothesis. If you show, in fact, that the prediction that you've made is not true, that it's wrong, then you can conclude that that hypothesis is not the reason for what you're seeing. Now, if you actually do the experiment and find out the predictions that you made are true, that doesn't mean you've proven anything. You've just got some evidence that it might be true. And if you do the experiment over and over and over and added variation to it, that ant can count, and you can't show that it's wrong, then there's more and more and more evidence, in fact, that it's likely to be true. So, in this, we're talking about a computer that won't start. It's not plugged in. It's got a battery. We think it's a dead battery, just like the car. So that's the observation, and we come up and we say, okay, we think that the dead battery is making the computer not be able to start. That's a hypothesis. One prediction is that if it was the battery and we plugged the computer in, that in fact it would start. Or we replace the battery. Okay. It still doesn't start after you plug it in, so we falsified that prediction. That's the actual test. So you know that wasn't the problem. Maybe the power button response is impaired, so you have to reboot your computer. That's a new hypothesis. You say if you press the button, let it reboot, it might actually come on. And you do that experiment by testing that prediction, and you find out, in fact, that it does. Now remember, this is just for, let's say, you have a problem with a computer, but in reality, let's say a computer maker wants to make sure they're selling people computers that work. So what would they do? They would take a lot of computers and test a subset of them compared to the whole population of computers. Okay? So not just one computer. But in this case, we're just trying to figure out what the hell's wrong with my computer. Okay. So here we go. We have an observation, or we have theory that people have proposed, and that we could perhaps test some of the predictions of those theories. The explanation is the actual hypothesis for why we observe what we observe. And it must be testable. Remember that guy Feynman in the black and white video that I showed, how he said the most important thing about science is that you can be wrong. What that means is you must be able to test an explanation that you can prove is wrong. In other words, the predictions that you made from that hypothesis fail. That's called falsifiable. If you can't do an experiment or something like that, then it's not a scientific hypothesis. Okay? So to test, you have to be able to show in fact that it is wrong. If it's not wrong, if you think you've got the answer, well, there's some evidence for it. The predictions, let's say, you make are correct. And then over time, it's a provisional acceptance, but it doesn't mean it's exactly true. So Newton came up with the theory of gravity in the 1660s. And that theory of gravity that made very specific hypotheses and predictions was the knowledge at that time that lasted for 300 years. And then along came a scientist by the name of Albert Einstein, and he started thinking about Einstein's theory, Newton's theory of gravity, and he came up with a new idea. And he came up with this theory of gravity. It was called general theory of relativity. It made very, very different predictions. Einstein's theory is what we currently believe, and scientists are still testing those predictions. Like Einstein predicted, his theory predicted, that there would be things called black holes with very, very strong gravitational fields. He also predicted that when two objects that were very massive came together, they would produce ripples in space-time called gravitational waves. That was recently tested, and it was upheld as well. So there's even more sense that Einstein was correct. But remember, Newton, he wasn't entirely wrong, but he wasn't right. The same thing might happen with Einstein as well. So you just have to realize that science is an ongoing process, and knowledge is provisional. That means it's not set in stone like dogma, like this is how it is. This is the evidence. That evidence suggests that it's this explanation. The explanation makes certain predictions. And that if new evidence were to come along, like Einstein's evidence and predictions that Newton's theory could not make, then we show that Newton's was provisional until new evidence came in, a new hypothesis came in, and it proved more robust than Einstein's. Okay, so here's a very simple, you've kind of seen this. You start with an observation. A hypothesis is strictly an explanation. A good hypothesis makes very specific predictions that can't be confused with something else. And then you set up an experiment to test those predictions. Now, if you find that you can't reject those predictions, if you can't falsify those predictions, there's some evidence that the hypothesis is true or might be true. But it's conditional. So there's a higher probability that your hypothesis might be true than before you did the test. If you actually show that your hypothesis and the predictions that it has are wrong, then you reject the hypothesis and you have to go back and come up with a new hypothesis. Okay, so we're going to talk about what constitutes a good experiment. First of all, we usually test experiments by things that we can change. Like with the ants, the variable was actually leg length, because the hypothesis the ants can count made the prediction that if we varied their leg length, they would not be able to find their way home because their pedometer inside their head, or nervous system, was being fooled. So, and that's all we wanted to test. We didn't want to test were the ants some other variable. Let's say some ants went out normally and other ants stayed in the nest and took care of the young, things like that. We're just interested in one thing, and that's the one variable that we manipulate. Now, do you think if you're taking part in a study or a new drug, and they set up two groups, and one group got the drug, and the other group didn't. Would you actually be able to tell if there was a difference? Now remember, both the individuals in each group know whether one got the drug and the other one, they didn't get a drug. They would know, right? Now, there's something called the placebo effect. Anybody know what a placebo effect is?
Placebo effect is when you're given something and because you keep telling yourself this should help me or this should do this, that you start to believe it whether or not it really does.
Right. Even if you don't believe it or not believe it consciously. And the placebo effect is very, very real. So the reason that we treat the subjects exactly equally is to control for the placebo effect. And if we're doing two different drugs, and we randomly pick people and assign one group getting drug, let's say it's a pill, the other group has to get the exact same pill. That way they don't know whether they actually got a drug or not. They might believe they got a drug. So this should control for the placebo effect, because you've treated both groups exactly the same. In other words, no variation except in the thing you're interested in. And what is that called when subjects don't know whether they got something or not? Blind. Exactly. So it means that the subjects are blind. Okay. Now, there's also, unfortunately, experimental bias or experimenter bias. Let's say I'm doing the test. And I've come up with this new drug, and I swear it's going to be a game changer. I know in my heart it's going to be a game changer. If the experiment is done, should I know which group gets the drug or not? No. Anybody yes? I don't? Okay. That means I've come up with a hypothesis, but who gets the drug and who doesn't, who gets the placebo, means that I don't know either. Because there's something called, there's all kinds of things that can happen when you're not treating things equally, whether it's subject or experimenter. And for experimenter, one of them is confirmation bias. Scientists want to believe what they believe. Now, most scientists aren't. Most scientists actually want to try to prove themselves wrong and not right. But you want to have a situation where the subjects don't know whether they got, let's say, the drug or not, and then the experimenter doesn't know either. So you can't get confirmation bias or some other bias. So like the estrogen thing that I was telling you about, well, there was a bias in the experimental design before they actually did the experiment. They just did a survey. And then, depending on how, then you want to find out, okay, yes, there's a difference, let's say in the drug. the person who gets the drug doesn't get a certain disease, the person who doesn't get the drug gets the disease, the one who gets the drug doesn't. Okay? But, remember we talked about sample size. So, to determine that, we have to do statistics. And when, let's say the election we just had, and you listen to pollsters go out and they ask a subset of the population a sample that they think is representative of the whole population, and let's say finds out how many individuals are voting for Trump and how many for Clinton. And they give a percentage. They don't just do that. They also give variation, plus or minus, let's say, 3%. That means that's the error that they accept. That out of 100 people, 3 might not actually be, let's say, telling the truth or something like that. And so usually they report the number of individuals in the survey that are used, 2,000 people were interviewed, and they'll give you some sort of variation around the mean, the average, whether they voted for one or the other. And that might be, hopefully it's a very small number, percentage. Okay. Now, I talked about random. The reason we do random assignment is we want to get a representative sample of the whole population. In other words, we don't want to pick the subjects and pick the ones that are more likely to show the effect than not. That's why we randomly pick them. And that way, there'll be a mix of, you might say, well, there's some criteria we want to control for age. Or we use age as a control variable. So that we can take away the effects of age and just look at, let's say, the treatment. Exercise level. So we have another variable that we control for. So we know it's not really because of exercise. The more things you can control for, the better. Usually now, most medical experiments, they control for age, they control for gender, but they didn't always used to, and it turns out like women usually have different symptoms for a heart attack than men do. Men are more likely to go drop dead. Women not so much. Okay. Now, control. The control is the group that doesn't get the actual treatment, like the drug, but they get something similar. Let's say get a pill that looks like the other pill, and they don't know whether they got the drug or not. That's the control group, and then the group that's actually getting the treatment, that's the manipulated group. Okay. So what's the name of the variable that's being manipulated? Huh? Anybody?
Did you say it was a control group?
No, because we're just talking about whether they got the control or not. So what's the name of that variable they can manipulate? Well, you could say the manipulated variable, we usually call it the independent variable. Okay, that means that this is the variable that we can change to actually test for the effect. In this case, let's say that one group got the drug, one didn't. What's the name of the response of the the variable we don't? We just measure it after the other, yeah. Dependent, exactly. That means we don't do anything to that. All we do is measure that. In this case, the number of people who are protected from the disease versus the number who aren't. Now, here's a question. Which is better in terms of determining the actual truth? Do a survey to ask who takes vitamin C and how often they catch colds, and see if there's a relationship between vitamin C and not getting or getting a cold. Do a survey and ask who takes vitamin C, then put the vitamin takers into one group and the rest into another, and then expose each group to the cold virus. In other words, try to give them a cold. Or you take a large group and randomly divide them into two groups. One takes vitamin C, the other takes a placebo for 2 weeks, and then you expose them to the virus and see who gets sick. What do you think? C. Okay? C is a real, honest-to-goodness, double-blind experiment, assuming first that the groups don't know who got what, and the experimenter who's doing it doesn't know either. Just so you know. Yeah?
Oh, so when you do a double blind, is there typically like a third party that determines who gets the placebo?
Yeah, you have, let's say you have someone use a random number generator, who gets what, and then you have some technician or whatever, and he doesn't know, but he's given either a pill for this group or a pill for that group.
So, in other words, they can still see who's been given what, they just can't determine who it is. They can't handpick, they have to assign numbers and pull them out of a hat. And
Yeah, but the subject shouldn't know, and then the experimenter who's actually running the experiment shouldn't actually know. They should just collect the data at the end and then analyze the data. And because they didn't know, they can't decide whether it's actually vitamin C or not unless there's a real effect. Okay? So that's a double-blind experiment. That's the kind that's the gold standard. So we've talked a little bit about bias, subject bias and volunteer bias. Now, sometimes you can't have blind subjects. And what I mean by that is the subjects actually know who got what. Like the ants. You can't give ants and treat them exactly the same way and then say, okay, this group doesn't know that they have their legs cut off. That's kind of obvious, right? So, you can't always do that. You can't always have a blind subject. Subjects who don't know which group they're in. And same with the experimenters. You set up something and obviously, well, the ant thing you could have done in terms of the experimenters, but even they would know, sure, because they would see. But let's say the experimenters, they get someone else to collect the data and then analyze it. So, do you tell the subjects which group they're in? No. What is it called when they don't know? Blind. How do you treat the control group? The same, or let's say you don't give them the drug? The same. Because you want to control for the placebo effect. If the control group knows they didn't get a drug, and they think the drug will actually work, and they think, well, I didn't get the drug, so I'll probably get sick, then you haven't controlled for the placebo effect. So you treat each of the treatment groups, including the control, exactly the same. Okay, so talked about confirmation bias. There also might be other kinds. Let's say you have a technician who's not very good at collecting data. Okay, now how do you control for that? Well, that can be a real problem. Now maybe you have a couple of different technicians. You find a couple, some are reliable, some aren't. You can actually create another variable control for that. And then double blind, that means subject and researcher don't know who got what. Okay. Now, I was talking to you about the estrogen replacement therapy. What we could do is we could have gone and talked to every woman in the United States and say, this is the population, and we get a census. We would actually get real numbers that are true. We wouldn't even need to do statistics. But usually we can't. Usually we have to take a smaller subsample, treat them exactly the same, and then determine whether there was an actual difference or not. And we use statistics to do that. So that's why we need a representative sample of the population and not one that's biased. And we can have descriptive statistics like the percentage of the population of the sample who is, or the population is male and the percentage who is female. Something like that. And then we can also use the data, if we know the sample size, to make inferences, which means, was the results actually significant? Did the effect that we imposed on the treatment groups, whether they got a drug or not, was that real, or was that actually just by chance alone? Okay, so the important thing is the sample size. The bigger sample of the population, the better. If, let's say, that drug allows people, it's a cancer drug, and it allows people to live an extra 3 months, versus another drug that actually cures people. Well, 3 months, if it was just 3 months, it would mean that the effect size is fairly small. Now, if you did enough people sample, you could actually show that it's significantly different. I mean, overall, the average lifespan of someone getting the drug, they live 3 months longer. Some will, of course, live less than that, some longer than that, but in general, they will average longer. So you want a large sample size, and if you can get a large effect size, that's great, but you can usually control for that. Because if you get a large enough sample size, you can still show that there is a significant effect. Now, sometimes you could actually design an experiment where you get a significant effect, but the effect actually has no real meaning on the population. So let's say an individual, the average individual, lives a week longer who got the drug and not. Well, it's basically about the same. In other words, the drug isn't very effective. Okay. Now, we have two groups, or let's just say we have 4 people. And each person flips a coin 10 times. The first person in group 1 got 6 heads, 4 tails, and then the second person, third, fourth, got 5, 5, 5. Two groups, the first one got 2 heads and 8 tails, 7 heads and 3 tails, and then 5 and 5. And then if you add the statistics on it, you'd sum and then do a bunch of stuff, and you'd find out that 21 and 19 is not significantly different than what you would expect, which is 50-50. Okay? Now, we know that if you got 5 heads and 5 tails, you would end up with 20 on the bottom. But you're more likely to get an outcome that represents what's really going on, 50% chance, the more samples you do, the more times you throw and flip the coin. So if you only do it 10 times, the difference between each, let's say you do it 10 times and you got 5 heads and 5 tails in one, and then you do it again and there's 7 heads. It means that each difference is in 10% increments. You've only done it 10 times. If you did it where an individual did it 1,000 times, the difference is that you're measuring is actually 1,000. And so you're much more likely to get, let's say, one group gets 510 heads, another person gets 498 heads, so you'll find that it's much more precise. Now, here's effect size with the vitamin C. The group on your left are the group that didn't get vitamin C. On the group on the right got 1,000 milligrams of vitamin C per day. And then on the left, the number of subjects was who got colds. It seems like, let's see, almost 90% who didn't get it got colds, and only 10% who did get vitamin C didn't get colds. But what if you only had 5 individuals in each group? Right? I mean, you don't have much power. It may seem like it's significant, but you would actually have to do real statistics on it to see whether that effect was that big. Now, it certainly looks like it, but it would be much better to have, let's say, 100 individuals in each group, or 1,000 individuals. The same with this.